All right, for our next award plenary talk, I'm honored to introduce to you the recipient of the 2019 Yentsch-Schindler Early Career Award, Dr. Rob Spencer. This award honors an early career aquatic scientist for outstanding and balanced contributions to research, science training, and broader societal issues. Rob's receiving the award for his contributions to our understanding of the biogeochemical processes involving the production, fate, and transport of organic matter in terrestrial, freshwater, and marine environments, and his projections on how anthropogenic impacts will affect these processes. Rob's an associate professor in the Department of Earth, Ocean, and Atmospheric Science at Florida State University. He received his PhD in biogeochemistry from the University of Newcastle in 2006, followed by a postdoc at University of California, Davis, and service as associate scientist at the Woods Hole Research Center in Massachusetts. So Rob's research on carbon cycling and the fate of organic matter in aquatic systems broadly aims to understand how climate and land use change impact the global carbon cycle. His research spans the fields of limnology, oceanography, soil science, and organic geochemistry, and he uses a diverse array of analytical and experimental techniques to address complex questions. Rob's really a pioneer in his insightful application of organic and isotopic tracers to some of the most difficult environments in the world to work, which also face the most pressing environmental impacts. He's worked on the glaciers of Alaska and the Tibetan Plateau to assess organic carbon storage and release from glaciers. He's worked on some of the Arctic's largest rivers to detect signatures of permafrost thaw, and in the Congo River Basin and the Amazon River Basin to assess land use impacts in tropical watersheds. And Rob's career thus far highlights his ability to bring together diverse information about ecosystems in a holistic view of how carbon dynamics are changing at the land-ocean interface, identifying the drivers of that change and what that means for the global carbon cycle. And this has led to an impressive publication record of more than 100 peer-reviewed citations, or publications, more than 100 citations, including more than 50 in the past 4 years. And beyond his remarkable research accomplishments, Rob is a generous and valued collaborator, and he invests considerable time in mentoring students at all levels. He's spent multiple summers in Siberia mentoring undergraduate students in aquatic research as part of the NSF-funded Polaris project. He's developed an innovative program to teach middle school students about aquatic science in the changing Arctic, and he's devoted to the success of his students and postdocs in his research group. So in summary, Rob exemplifies the intent of this award by excelling in all aspects of his career, from research to mentoring and outreach. So please join me in congratulating Rob Spencer.
Thank you, Kim, for that truly wonderful introduction. I'd like to start by saying it's a true honor to be here receiving this award this year. And it makes you step back and you realize that it's not just you receiving this award, it's everyone really who I collaborate with. And I'd like to start with a few thank yous. First of all, the people who took time out of their busy schedules in the award selection committee and the people that took time to nominate me, as well as the ASLO board and staff for putting on another truly wonderful conference this week here in Puerto Rico. And then unfortunately, I only have 15 minutes because I could spend hours up here thanking many, many mentors, collaborators, and colleagues that have helped me get to be in this position today. And I'd just like to say you all know who you are. I hope you all truly know how much I appreciate everything you've done for me in my research career. And a big thank you, and I sincerely am very grateful for everything you've done for me in my career. As I was thinking about the theme of today's talk and what I'd like to talk to you about, ASLO actually did me a huge favor. As part of the broader outreach around these awards, they sent me a number of questions, and These questions are really nice questions that are aimed at kind of what do I do as an early career researcher and how do I address some important issues. And so I thought I'd share the abridged version of those questions with you for my presentation today and then hopefully answer them. And the first question was, what advice would you offer to students and early career researchers about successfully publishing papers? And why is your research important? And what are some of the greatest benefits and challenges of engaging in international research collaborations? And so I'll take 2 and 3 together, and they're kind of the running theme when I'm talking about a lot of the research in my presentation. And so I'd like to start with number 1, which is basically, why is publishing important? And as I was thinking about this, and I thought about this for a long time, I went all the way back to my graduate school experience, and I was very fortunate. I actually had 4 advisors in graduate school. I was part of a new program at the time in the UK that was trying to bring scientists from different backgrounds together to work across those backgrounds. And I actually, as part of that, had advisors at a number of universities. And so I had Greg Cowie at the University of Edinburgh, who first kind of introduced me to biomarker research, something we continue to do today. Gunther Ewer at the University of Newcastle, who's kind of the consummate, my door is always open, come and talk to me, professor. And Andy Baker, also at the University of Newcastle at that point, who first introduced me to optical measurements like absorbance and fluorescence and the power that they could give you as an aquatic scientist. But really, I ended up in grad school in the first place due to Robert Still Goddard. He was just a wonderfully inspirational scientist and a mentor. And I worked in his lab as an undergraduate. I worked for a number of summers there, and I worked for him for a summer when I first graduated. And his enthusiasm for the science he was doing was really something that caught me. And I thought, maybe this is something I can do with my life. And so I chose to go to graduate school. I there first made my first collaboration, Aaron Stubbins, somebody who I still continue to work with to this day. He was a graduate student a few years senior to me in the graduate group there. And that's also a very useful thing to have, because then you have that person on a day-to-day basis you can ask those questions to that you might not feel so comfortable asking your advisor about. And I'm very grateful that Aaron still continues to work with me now for nearly 20 years or so. And so I had all this support, but unfortunately, the actual research I was trying to do wasn't going very well. The analytical side of things wasn't really working out. And I was actually at that point thinking about leaving graduate school because it didn't seem to be what I expected it to be. And then 2 things happened, which is really what started me on the road to being stood here today. First of all, I read this paper. What happens to terrestrial organic matter in the ocean? So this paper's over 20 years old now. It's been extensively cited. And it's just a wonderful example of, in my case, how a publication can make an impact on you. Because the problems I was having with my data set, I started thinking about them in terms of answering this question, which wasn't really the goal of my research. It's a wonderfully written paper. It has a clear statement. It basically describes the research at that current time point 20 years ago, and then it outlines what we can do going forward to try and answer this question. And so this paper really sort of caught my attention. Shortly thereafter, I came to my first ASLO meeting, which was the Aquatic Sciences meeting in 2003 in Salt Lake City. So I was a young graduate student at this point, and I gave my first presentation, which was a truly terrifying experience at that point as well, to stand up in front of all your peers and senior colleagues. And at the end of it, I was asked a question by one of the authors of that paper, and he came up to me after the talk, he took the time and said, I enjoyed your talk, I'd like to talk about a few things, can we go for lunch? And so he invited me to lunch, started introducing me to other scientists that were there, and a couple of things came away from that lunch. First of all, he explained to me that some of the analytical issues I'd been having, I wasn't the only one. Right? A lot of other people had gone down these roads and had faced similar issues. And so it was important for my confidence. It gave me that back. And secondly, it really helped me sort of frame my data around this question about what happens to terrestrial organic matter in the ocean. And so if you're an early career researcher listening to me out there, make use of these meetings. They are incredibly important. Go up to people. Everyone in this room will be happy to talk to you about their research. Okay, so don't be shy. Go and talk to people. That's what we're here for as a community. So I then moved forward. I graduated from my PhD, and I was still somewhat obsessed with this question of the fate of terrestrial organic matter in the ocean. And at that point, I met someone who was equally obsessed in my postdoc advisor, which was Peter Herness at the University of California, Davis. And Peter was really interested in trying to reassess some of the biomarker data. And I'll talk a little bit about that on the next slide. And shortly after meeting Peter, I also met George Aitken at the US Geological Survey. Again, at a meeting like this, I met somebody who then became a very important mentor for me. And George really did a couple of things for me. First, he opened the doors to working in the Arctic, working in Alaska with the US Geological Survey. And secondly, generally just collaborating with the US Geological Survey, which is something I still do extensively to this day, and allows for us to make sort of truly global projections by working with fantastic colleagues who have lots of data from all across the United States. After I met George, I also then met Max Holmes, who similarly opened up all his contacts, his connections to help me start doing work on Arctic research, but this time in Siberia. And so with these three people in place, these three senior colleagues mentoring me, we set about reassessing the biomarker data to look at the fate of terrestrial organic matter and really trying to improve estimates for its removal and improve flux estimates of terrestrial organic matter to the ocean. And so this is important. So if you look at some data, this is a classic hydrograph from the Yukon River. You can see that nice big pulse in spring discharge in late May, early June. And concurrent with that, we see elevated dissolved organic carbon concentrations and lignin concentrations. And so lignin's a terrestrial biomarker. We also see elevated lignin carbon normalized yield, so we have a greater proportion of our organic matter coming from terrestrial sources at that time of peak discharge. And along with that, we see elevated acid-aldehyde ratios in the vanillyl phenols, which historically we would interpret to mean that this material is highly degraded, it's recalcitrant, if you will, and it's going to be very stable in the environment. But along with other groups at this time, what we were doing is starting to make multiple measurements of our organic matter. So we're bringing multiple techniques together. And concurrent with the lignin data, we were finding that the dissolved organic carbon is also younger, so it's fresher, it's more biologically labile, and it was very photoreactive. And so all this leads to the fact that that 60 to 70% of your annual lignin load that's been exported in that 6 to 7 week window around spring freshet is actually much more reactive in the environment than we historically thought. And it really is a case, as Peter likes to say, of molecular trickery. So at that time of maximum DOM export, the molecular signature is telling us one thing because we were using it in a way that was appropriate in the particulate phase, but really isn't when we're looking at dissolved organic matter. We were also interested in increasing flux estimates, and one thing we turned to here was very simple measurements like chromophoric dissolved organic matter. You know, we can measure these on small volumes of water. It's very cheap to do. We can also do it in situ, and we can do it via remote sensing techniques. And so this is looking at data across the big 6 Arctic rivers, and if we can set up strong relationships like those shown here between the colored dissolved organic matter and dissolved organic carbon concentrations and lignin phenol concentrations, then we can take any CDOM measurement we have and use it to improve our flux estimates. And so we started making a lot of high-resolution measurements across that highly dynamic spring freshet period. And when we did that, we found that we were increasing the DOC flux estimate by about 30 to 40%, and the lignin flux estimate doubled at that time. And that's very important because it means that that terrestrial DOC, the dissolved organic carbon, is then transiting through the Arctic Ocean much faster if that's more representative of current Arctic riverine export. And so that's another way that really shows that this Arctic riverine dissolved organic matter is more reactive than previously thought. And on any riverine system where we've taken this approach, we always come away with a similar answer. So at this point, I'd like to pause for a second and say, I was very fortunate then to be offered, if you like, a faculty type position and to move forward and start building my own laboratory. And there's a whole host of people from undergraduates, graduate students, postdocs, visiting scientists from all around the planet who've come and visited in the last 8 or 9 years or so. Thank you very much. This talk, you know, you're all part of this. And really now you guys are the engine of the research that's happening in the lab, and nothing makes me happier than seeing you guys go on to be successful with your own careers. And so if you're on one of these 2 slides, I'm very proud of you. And so we're working all across the planet now. We still have a big focus on land-to-ocean fluxes, but we've also started working on how humans are impacting the carbon cycle, particularly in aquatic ecosystems. And so as Kim nicely introduced, we're working in basins from the Amazon and Congo all across the big Arctic rivers and then closer to home as well. And so for the remainder of this talk, I'm going to talk a little bit about data from the Congo and also up here in the Kolyma River Basin in Siberia. So I'm sure most people are aware that the Arctic is changing faster than anywhere else on the planet. And one of the big concerns we have is what will happen to all that organic carbon that's stored away, frozen in permafrost soils. There's over twice as much organic carbon frozen in permafrost soils in the Arctic than there currently is in the atmosphere. So as this material thaws, is it just going to be mobilized and go out to the ocean from one long-term store to another, or are microbes going to utilize it and it's going to go back to the atmosphere as greenhouse gases? And so just highlights of some data from Arctic systems here in Siberia. The blue lines show bioincubation data of dissolved organic carbon over 28 days. And what you see is rapid loss of that dissolved organic carbon in these headwater streams, greater than 50% loss in less than a week. And importantly, the radiocarbon age isn't changing through those bioincubations. And so we know that the microbes are truly utilizing that old fraction of the carbon. And if you compare that to the Coloma River, it's dominated by modern dissolved organic carbon, and it shows very small losses. It's much more stable. And so this has big ramifications for our projections moving forward for the fate of this material. And we've also been interested in terms of which fraction of the organic matter pool are the microbes utilizing, and is there some tracer in there that we can use to look at permafrost carbon mobilization into the future. And so increasingly we've turned to ultra-high resolution mass spec techniques. This just shows a Van Crevelen plot with O to C on the x-axis and H to C on the y, and each individual dot represents an assigned molecular formula. And what it's showing is the change in a 28-day bioincubation of permafrost thaw material. So the blue dots are ones that are being utilized, the red dots are those that are being produced. And you can see the selective loss of the aliphatic molecules, and you see production in this classic area in Van Crevelen space, the so-called nicely termed island of stability that we find pretty much everywhere in riverine and marine environments. And so the microbes are actually taking that unique signature of permafrost thaw material and turning it into DOM that we find in other riverine and marine systems, and making it very, very hard to detect unless we're looking in these small headwater systems. So the other side of what we do is increasingly we're working in the tropics. This photo is from close to the border with Rwanda, you're in DR Congo here, and 20 years ago this was all pristine forest in this ecosystem, okay? So this is rapidly being deforested, converted to agriculture, simply due to population pressure on the landscape. So we've set up a nice space for time gradient in the region where we have watersheds from 100% pristine in the Kahuzi-Biega National Park all the way through to completely deforested catchments. So we can start making projections of how deforestation and agricultural impacts in the region will occur moving forward. And so on all these graphs I'm going to show you on the x-axis, the x is the fraction deforested. And if you look at the 13C and 14C signature, you can see it varies greatly with deforestation extent. And those deforested streams are becoming about 6 parts per mil more enriched than what we see in the pristine forested streams. And at the same time, those deforested sites are also showing a shift in radiocarbon age from modern to about 2,500 years before present. So we're seeing a shift from a modern C3 source to an aged microbial-looking source. And so if we go to one of the pristine sites and take a soil core, what we see through that is if you look at the litter layers, it looks exactly like what we see in those forest streams. It's modern, and it has a C3 signature. As we move down that soil core profile, it becomes more enriched in 13C and depleted in radiocarbon with depth. And so deforestation is tracking that soil organic carbon isotopic signature with depth. And basically, the sites that are draining from the deforested landscape are showing that your organic material is sourced from about 0.5 meter depth, which really just tells you you've lost 0.5 meter of topsoils due to that conversion to agriculture in less than about 20 years. So it shows you the ramifications for the carbon cycle moving forward. We're also, as we are in the Arctic, interested in the fate of this material, so some biolability data here. Again, it shows that you see significantly higher biolability in the deforested sites than in the pristine sites. And interestingly, that biolability is negatively correlated to the radiocarbon age, so that the older material is relatively more biolabile. When you think about it, you'd think fresh things are often more biolabile. But in these systems that are being impacted by humans, this is a picture we keep seeing time and time again. And it's, if you like, additional mobilization of aged carbon by human activities with then clear ramifications for the carbon cycle. So one of the wonderful things about working in Congo is everywhere we go, people are interested in what we're doing. And so you can see some images here of just sort of typical field work at some of our sites. And what we've really been trying to do in recent years is not just tell people, this is why we're doing this science, this is why it's important, but to heavily engage with them and to really engender that local scientists are going to be the next generation of scientists doing research in this very important region that's going through rapid change. And we've taken that approach all throughout where we're working. You can see examples here from working with indigenous communities in the Arctic, and really building that through traditional education. You can see K through 12 education there, and taking things like permafrost soils into the classroom so that young students can have a hands-on experience with what is essentially frozen dirt. But once you've ever smelt permafrost, it's something that smells with you. It's something that sticks with you. And so it's something that will hopefully build that next generation of not just aquatic scientists, but also environmental scientists. And that's also been recently supported by ASLO. In the last photo there, you can see an example from an inner city school in Sao Paulo, Brazil, where we're asking young students, what is the Amazon to you? These students have never been to the Amazon. A lot of them are drawing images of what they think the Amazon is, but it's just trying to get them to connect with their important aquatic environments and the important ecosystems within their countries. So to summarize a few final thoughts, it's incredibly cliched, I apologize for it, but it's true. It's why I'm stood here today. Do what you love and you'll never work a day in your life. I think I'm truly lucky to have this as my career. And enthusiasm is contagious. I mentioned this at the start, kind of what got me down the graduate school path. I don't come from a background where anyone in my family had gone to graduate school. Is that I saw somebody who was truly enjoying what they were doing and they were making a difference. And that really captures people's imagination. If you're not enthusiastic about your own science, it's hard to engage with people, okay? I've talked a lot about research at the boundaries today, land, ocean, biology, chemistry, soil to aquatic interface. And to me, there are a lot of really exciting questions at the boundaries, and there's an increasing need to be well-versed in different traditional disciplines. It really helps you, even if nothing else, talk across those disciplines, and it certainly helps form strong, creative, functioning, collaborative teams as well. In terms of scientific training, I think more than ever, everyone in the room will agree with me that we're facing more threats to science than we've seen before. There's more people just generally disregarding what scientific knowledge is telling them. And funding's increasingly challenging, so I encourage you all to support each other. This is a community. And if you're set out there today and you're successful, your career's going well, give back. Ask who it was that helped you get to where you are today. If it works for you, there's a good chance it will also likely work for other people as well. And then I talked a little bit today about manuscripts and their importance, but I always ask my students this: how many people actually read your manuscripts? All right? It's a very small number. Most of them are locked behind paywalls. They use scientific jargon that's very hard for the general public to access. And so please do not be afraid of the media. Some people have very dim views about scientists engaging with the media. I strongly encourage you to go and do it, if for no other reason that if you do not, some other voice will fill that void. And that is not necessarily a voice that we want filling that void. And then finally, all of you sat in this room are truly ambassadors for us as aquatic scientists. So when you're out doing field work, when you're talking to your families, your friends, engage and excite people when you have the opportunity. And I'd like to say thank you once again for your time and this truly fantastic honor, and thanks some of the agencies that have supported my research in recent years. Thank you.